70% depletion of JAK1, and so on and so forth. Because JAK1 was down.
Our next student presenter is from CHS, and continuing along the same kind of theme. They're also looking at DNA damage. So it's Anand Singh, and his title is Investigating the Role of Microhomologies in Double-Strand DNA Break Repair. And I think Dr. Damon Meyer is the fact mentor here. So thank you. Please go ahead.
All right, thank you for that introduction. Good morning, everyone. Thank you all for being here virtually. My name is Ishaq Aslam, and I'm from the California North State University College of Health Sciences. And today I'm going to be presenting our research on investigating the role of microhomologies in double-stranded DNA break repair. And so I'm a part of Dr. Meyer's research group at CHS, and so we've worked on this project in the last year. And so unfortunately, my lab partner Anand Singh could not be here with us today, but not to worry, I'll do my best to present the research that we both worked on together in the past year. So jumping right into it, hang on. What exactly are double-stranded breaks? So double-stranded breaks are lesions in cells' DNA that result when both strands of the DNA double helix become severed. And so these double-stranded breaks, which are also known as DSBs, can result from a multitude of factors, including endogenous damage through normal metabolic activities, such as the production of reactive oxidation species, and they can also occur from exogenous damage, which basically comes from the environment. And this can come from UV radiation from the sun, as well as other chemical agents in the environment. And so DSBs are also associated with cell division, with up to 50 breaks occurring per cell each replicative cycle. And so this can result in as many as 1 million molecular lesions per cell each day. And so if these lesions are left unrepaired, the cell can enter one of three states, all of which are not too great for the cell. The first state is known as programmed cell death, also known as apoptosis. The second is cell senescence, which is basically irreversible cell dormancy. And then finally, and in my opinion probably the worst one, is the formation of malignant tumors. And so the way these tumors form is that when both alleles of a tumor suppressor gene become inactivated because of a double-stranded break. And so this leads to the formation of cancerous cells and cancerous tissues, and it's all in accordance with the two-hit hypothesis when those two alleles get knocked out. And so in order to repair these double-stranded breaks, nature has given most eukaryotic cells and organisms three main repair pathways to utilize. The first of which is known as non-homologous end joining, shortened to NHEJ. And this is basically when the two ends of the DNA are directly joined together via DNA ligase 4. The second is known as homologous recombination, abbreviated to HR. And this is basically when a homologous chromosome or sister chromatid is used as a template for DNA repair, and it utilizes a similar mechanism that's found in crossing over, which happens during meiosis. And finally, the last mechanism and the focus of this research and this presentation is known as microhomology-mediated end joining, abbreviated to MMEJ. And so MMEJ is special because it utilizes microhomologies, which are basically 5 to 25 base pair sequences that are found in the genome, and these sites serve as initiation points for DNA repair. And so similar to homologous recombination, MMEJ utilizes a homologous chromosome as a template for DNA repair. However, homologous recombination does not involve those microhomologies in double-stranded break repair. And so our study focused on MMEJ because, unlike the other two repair pathways, NHEJ and HR, that I had mentioned, MMEJ is known to be a mutagenic pathway, and it's associated with both the progression and proliferation of cancers. And so with one of the main differences in this pathway being the utilization of those microhomologies, the purpose of our research was to investigate how both the length and continuity of these microhomologies, these repetitive sequences, influences the effectiveness of double-stranded break repair via MMEJ. And so our study utilized Saccharomyces cerevisiae, which is basically Baker's yeast. It's known as Baker's yeast commonly. And we utilize these as our model organisms for a number of reasons. The first is that they are eukaryotic organisms, meaning that their DNA is packaged into chromosomes, and they also utilize similar DNA repair pathways as us humans. So the model organisms, it's a good model of organisms for humans. The second is that they're relatively easy to modify in terms of their genomes, and from that it's relatively easy to observe their genotypes and phenotypes through the use of selective media, which I'll be going into how we did that in the methodology. And thirdly, they're not biohazardous, they're relatively safe to use and easy to manipulate in the lab, so they just make it kind of easy to work with. So now kind of delving into the mechanism involved in MMEJ. So I'm referring to the figure here on the right. So in step 1, a double-stranded break occurs, and the chromosome basically aligns with its intact homologue, which is the chromosomes that are shown in red. And the formation of what is formed as a D loop occurs in step 2. And this is basically where the red intact homologous chromosome invades at that 3' end of the broken strand. And it's at this end where the invading homologous strand basically utilizes the microhomologies at the end, at that 3' end. And this serves as an initiation point for DNA repair. And so it utilizes that red homologous chromosome as a template for DNA repair. And so for our study, what we're doing is that we're changing both the lengths and continuities of these microhomologies, which are these like blue base pairs shown here, in order to observe the effects of MMEJ repair on double-stranded breaks. And so what we expect to see is that as the length increases and the continuity of these microhomologies remains constant and unchanged, the greater the recombination frequency, that is, the greater the repair of the double-stranded break via MMEJ will be observed. And so for our study, we obtained different strains of yeast that had varying lengths of continuity and continuities of microhomologies that were centered around the His3 gene on chromosome 15. And so this is basically where we're inducing the double-stranded break. And so the reason why we chose the His3 gene is because it's located centrally in the middle of the chromosome 15, and it's not too close to the centromere or telomere, which is important because it's away from all that condensed DNA. And so from there, it's relatively straightforward and easy to observe and place genetic markers on either side of that gene. And so for the experimental groups that we're testing this on, they varied in both length with 16 base pairs, 20 base pairs, and 25 base pairs, as well as continuities, where mismatches were introduced at various locations on the microhomologies. And so the control for our experiment was a strain of yeast with a complete and intact homology of 311 base pairs, and that would serve as our baseline for the comparison of results. And so now kind of getting into the methodology, how we conducted this experiment. So it incorporates a multitude of steps, careful tracking, and a bit of knowledge about Mendelian genetics. And so in order to get our desired genotype, which I had shown previously, in which we had those genetic markers centered around the His3 gene, we needed to conduct test crosses with yeast and basically analyze those crosses. And so with yeast, crosses can only really be done between alpha and A haploid individuals on YPD media. And YPD media is basically media that's beneficial for yeast to grow and multiply in. So this top left image here shows that one of the many crosses that we conducted throughout our time. And so in this instance, a cross was done between a trip alpha and a LU-A haploid individual in order to obtain a diploid genotype with both the trip and LU genetic markers. And so in order to specifically select for that yeast strain with that specific genotypes, the results from this top left cross would have to be plated onto media that was lacking in tryptophan and leucine, and those strains that had the desired genotype that we wanted would display that phenotype by being able to grow on that media that's lacking in leucine and tryptophan. And so going from diploid to haploid organisms was also necessary in order to obtain desired genotypes of either alpha or A haploid individuals which contain only one copy of an allele. And so this was basically done by plating diploid yeast onto SD- media, which is media that's basically lacking in all nutrients that forces yeast to undergo sporulation and thus undergo meiosis. And this is how you obtain the alpha and A individuals with your specific genotypes. And then again, the way that you confirm these specific genotypes was by replica plating them, the haploid individuals onto selected media, and then subsequently determining the results about which grew and which didn't via tetrad analysis. And so in order to determine whether you had an alpha or an A individual for a future diploid cross, mating type tests were conducted. So actually, at one point of our research, Mendelian genetics was definitely not in our favor because we had obtained only alpha individuals of a desired genotype, and we just needed one A individual with our desired genotype. So we had to kind of backtrack and reperform all of our crosses to get an A individual. And it kind of just goes to show that in research, not everything really goes the way you have it written down in your protocol, but you just have to adapt, improvise, and keep on progressing in order to continue the project. So This image and table kind of depicts the various genetic markers and aspects of our desired yeast strain that we conducted crosses to create. And this also shows how we induced the double-stranded break in order to observe MMEJ repair. And so, as I had mentioned previously, the HIS3 gene is located at the center of chromosome 15, and the end of that gene actually encodes for an HO cut site, which is where the double-stranded break will be induced via an HO endonuclease. And so on either side of the gene, we have the MET22 TRP1 on the left and the REV1 URA3+ on the right, and these serve as genetic markers and monitor crossing over events. The GAL-HO is basically what induces the double-stranded break on the HO cut site by the HIS3 delta 3' gene, and the promoter region of this gene basically becomes activated in the presence of galactose. And so once this happens, this gene basically expresses the endonuclease that creates that double-stranded break. And so the HIS3 delta 5' gene shown here is basically the homologue for the HIS3 delta 3' gene, and this is basically what's going to be serving as a template for the repair of the broken DNA strand. And so once the double-stranded break was performed by basically inoculating this specific yeast strain into galactose media and then activating that endonuclease, the yeast were then incubated onto YPD plates. And the number of cells that were able to grow on that plates, the viable cells, were counted. And then these were also plated onto SD-HIS plates in order to determine the number of cells that were able to grow in media lacking in histidine, which basically meant that if their cells were able to successfully grow, they were able to repair that double-stranded break via MMEJ. And so, this is our data that depicts the MMEJ recombination frequency for each of the various strains of differing lengths and continuities for the microhomologies. And so the way we calculated recombination frequency was basically by dividing the number of histidine-producing cells by the number of viable cells on the YPD plate. And so these error bars here are actually 95% confidence intervals, meaning that when they do not overlap with each other, the results that we obtained are statistically significant. And so looking at the data, you can see generally as the length of the microhomology increases, the number of viable cells and the cells that were able to undergo recombination frequency also increases, with our most viable cells coming from the longest homologies in the 300s, and the least number of viable cells coming from the smallest microhomologies with the 16 base pairs. In fact, you can see that there's actually a 55-fold difference when you're comparing the recombination frequency between the 16 base pair microhomology and the 25 base pair microhomology, and it shows just a 9 base pair difference, but that's how significant it is. And so in terms of how the continuity affects microhomologies, it can be seen that when a mismatch is introduced, there doesn't really appear to be a significant difference in the recombination frequencies when you're comparing a mismatched microhomology with its counterpart, which had a complete and intact unchanged microhomology, even when they are the same length. And so it appears that that with the length of the microhomology has a more significant impact on double-stranded break repair via MMEJ compared to the continuities of microhomologies. So, why is understanding the factors that affect DNA repair mechanisms important? So as I had mentioned previously, MMEJ is a mutagenic pathway. So studies have shown that microhomologies tend to play a critical role in initiating chromosomal rearrangements. And these rearrangements include inversions, duplications, and translocations. And so these chromosomal rearrangements are typical hallmarks of cancer and are bioindicators of the development and progression of cancer. In fact, cancers appear to almost exclusively rely on the MMEJ repair pathway as compared to the other DNA repair mechanisms, non-homologous end joining and HR, showing how MMEJ is a driving mechanism for the proliferation of cancerous cells. And so cancer cells, as you know, are constantly replicating and dividing, needing to repair those double-stranded breaks, and so they utilize MMEJ. So it only highlights the importance of studying the critical factors and players in MMEJ, because it's essential in understanding how cancers develop and progress. And so for future studies, we would like to further investigate these pathways by looking at the various enzymes and cellular machinery, such as RAD51 and POL4, and their functions in the repair of double-stranded break. Little is known about their mechanisms and what roles they play. However, they seem to play some role in DNA repair pathways, not just for MMEJ, but also homologous for recombination and non-homologous end joining as well. And so continuing down this path and looking at all these different factors could lead to the development of potential therapeutic targets for cancers, which basically target and utilize DNA repair mechanisms. And so yeast, being eukaryotic organisms, they share a lot of analogous machinery and mechanisms for DNA repair as us humans. So future studies with yeast that are safe and easy to do have relevant and significant impacts for human research and treatments. And so with that, I'd like to thank Dr. Damon Meyer for this. I hope you all were able to gain something. Oh. And I'd also thank my lab partner, Armin Singh, and the rest of our lab research group, Team Yeast at CNU CHS, without whom we could not have obtained all our data. Thank you all, and I'd be happy to take any questions.
Thank you. That was a very nice presentation, and what an elegant system you guys have. That's very nice. I can already see that you probably would want to collaborate with Dr. Shi from College of Medicine.
Yeah.
You two could definitely cook something up together.
Yeah.
So do we have any questions?
Hi, Ashok. I have a question.
Yeah.
I also want to agree with Dr. Vinal. I think this is a very elegant system. And actually, it kind of has implications for the talk just before that.
Mm-hmm.
CA, of course, is involved in double-strand DNA break repair. And I wondered whether you had anything to say about that, or whether anyone has done any, genomic look for potential targets based on something like microhomology repair, because that's really, what the kind of current treatment is for BRCA associated cancers, of course, is PARP inhibitors. So, I mean, I wondered whether, and I'm kind of going back to this idea, this is such an elegant system, whether there's any way to clone in candidate genes that are being repaired, for example, in a yeast system for a really quick screen.
Yeah, that's a great question. So, for most DNA repair mechanisms, HR is the preferred pathway because it's the most accurate. And so MMEJ occurs in about 10 to 20% of the time, but then in cancerous cells, it's kind of overexpressed. And the reason why that is because PARP is upregulated because of that. And so because of that, HR goes down, homologous recombination goes down, and the cells start using MMEJ, and which is more mutagenic. So like you were saying, PARP inhibitors are one of the kind of therapeutic targets because of that. And I think they've recently been approved in conjunction to work with chemotherapy. But a lot of the cellular machinery that are used in yeast are very similar to the ones that are used in humans. So there are definitely more therapeutic. There are definitely more enzymes that we could look at to conduct experiments that you're kind of referring to there.
Very good. Thank you.
Very good. Any other questions? So I'm just curious. So what are some of the proteins that are required for MMEJ? I'm not familiar with that pathway. So.
Oh, yeah, of course. So there are a number of enzymes that are incorporated for MMEJ, and so they have different names in yeast and versus humans. So humans have about 14 different DNA polymerases that are involved in DNA repair, and yeast have about 4 to 7. So there are a couple. So POL4 is one of the main ones that are used in yeast, and it has an analogous polymerase that are found in humans. I think it's POL theta. So there are quite a number of polymerases there.
I wonder if any of those were found by the previous presenters, if that was in their screen.
Perhaps.
Maybe.
Yeah, actually, yeah.
Oh, great. Okay, well, if there's not any more questions, I think we can wrap up for the oral presentations for this morning. And thanks to everyone for staying on time. We're actually a minute early. So now we're going to move to the